everyone, and welcome to our workshop here, the CDDF workshop on dose optimization in early oncology drug development. I'd like to welcome everyone here in Amsterdam on a very beautiful day outside in the harbor, and the many more people who join us online. You are very welcome as well, and please participate actively from the start by sending us questions. Back. Good. Let's look into what we are going to see. First, who are we? What is the CDDF? It's a non-profit organization registered in Belgium that provides a neutral platform to create a dialogue, collaboration, exchange between all the relevant stakeholders involved in cancer drug development. We bring together a cancer drug development community from academia, pharmaceutical industry, regulatory authorities, policymakers, patient advocacy groups to accelerate and improve development of drugs against cancer. And we are empowering patient involvement in this. We think it is very important to go to the most important questions that are on the edge of what we know, of what we have already established, and try to push further. And I think the question of dose optimization is a very good example for this. Please visit our website, www.cddf.org, to see more and to access our materials. I would like to give a very brief introduction. So I'm a physician. I spent most of my work in the clinic in treating hematological malignancies, acute leukemias, and that means 7+3 high-dose cytarabine chemotherapy. The more, the better. You learn to optimize dose intensity to obtain the cure rates that are obtainable in AML, especially 20 or 30 years ago. I don't want to calculate how long that was when I started. And these curves here show you, to your left, show you that the toxicity and efficacy curves of these drugs are practically in parallel, very steep. And so developing the doses of these drugs by determining the maximal tolerated dose, by determining toxicity. Thank you. I brought an allergy with me, so please bear with me. By determining the maximum tolerated dose, the MTD, by measuring dose-limiting toxicity was the way to go. And of course, with chemotherapy given over 5 days or a week or so, toxicity occurred early. In the first 2 weeks, maximum toxicity, and then patient recovered from that. So that was easy to measure. So in a way, the MTD paradigm, also not knowing better and not having clear measured single molecular targets for these drugs, was the thing to go, and it had its measure of success. However, lately we have made progress in the sense that most of the drugs we now approve in cancer are molecular targeted agents. And these follow different kinetics. Once the target is fully engaged, further dose increases are very unlikely to increase the effect, however, quite likely to increase toxicity, especially off-target toxicity. So here, and the curve to the right shows you an example of that. Here you clearly see that the paradigm of dose finding should change. And the same is true for other agents, so antibody drug conjugates or immune oncology agents, not even speaking of CAR T cells or more complicated products, need their own methodology of dose finding. And another very important part is the measurement of toxicity. Most of the treatments we give now are given for a long period. Again, my early experiences treating AML patients, we would give 1 or 2 induction cycles, then up to 1 or 4 consolidation cycles, and then it's over. And now most cancer treatments are given for a long period or until progression. So it's very important how much of the toxicity occurs after the first course, after the end of the DLT period. And here are just two examples for fatigue and nausea. These curves show you how much of it occurs later than the early period. But it also shows that the measurement by clinician-reported tools is insufficient, at least for some of the toxicities that are very important to patients. And other toxicities include diarrhea, include neuropathy, other things that take a long period, a cumulative buildup, that are not adequately addressed. And not measuring that toxicity and finding the right dose often leads to doses that are too high, that cannot be tolerated for long enough, and therefore quite probably reduce the clinical benefit that the patient could have from the treatment. This is an example, a curve of when the main immune-related adverse events occur with immunotherapies, checkpoint inhibitors, PD-1 or CTLA-4. And you see here as well, there is a distinct pattern of late occurrence that would not be captured in a DLT-driven dose finding. So that's enough. You will hear much more from much more competent speakers than me. My next step is to introduce the program committee. So first here, Katrin Rupala, our co-chair and very active member of the program committee. And there is also Harald and Chun-Se, who are here in the room. You will see them soon sharing a session. And they have been also very active over a period of, I think now 9 to 10 months, where we have been meeting and developing the program. So many thanks to all of you. The workshop overview is pretty straightforward. The first session looks into the challenges of dose optimization. The second looks into Project Optimus and FDA regulatory initiatives. And the third one looks into what are solutions, methodological approaches. Tomorrow, we will start at 10:30 with a kind of a compromise between European timing and US. At least those on the East Coast might find it possible to attend the first session here. And we start with options for the path forward, very interesting talks about safety and toxicity, patient-reported outcomes, industry perspective, and also case studies. And then we try to wrap it up in a panel discussion that is aimed at kind of fixing, really writing down the next steps that we should do. What should be done individually, what should be done in collaboration. And we intend to publish a white paper with these results. What are the meeting objectives? Obviously, many of the things that I've just said and others, of course, have put forward much more elegantly are quite clear. But what are the challenges? If you endeavor on that path, what makes it difficult? So we want to look into that. We want to discuss the opportunities. How do we do that? How is really dose selection done in different cases? What are the statistical methods? What can preclinical models contribute? What can biomarkers contribute? How to use PROs, how to use different kinds of safety reporting. All of that will be very important. Exploring these new methodologies must somehow then be translated into a dose selection. And that is another important point for discussion. How can we formulate, that's our main objective, how can we formulate a dose selection strategy in the future? How can it be implemented in early drug development? And how can we do this with considering all relevant stakeholders' views, including the patient voice, including the advice that comes from the regulators, including, obviously, the many pressures the pharmaceutical industry is under. So, how can you participate? Those in the room here, obviously, can raise their hand, go up to the microphone, and make a question, but you can also, at the same time, join us online, which we will recommend you to do for the audience interactive questions that will come in a minute. You will hear about it then. If you follow us online, you will be already connected via the software platform, and there you find an opportunity to send questions that should be addressed here in the discussion. Please do so. We have an iPad here in front, and so do all the other chairs, to see what questions are coming, and we will make sure that as many as possible will be answered. At the same time, there is a stream of quality messages summarizing the talks and also predicting what is coming up on our social media. So please follow us there. LinkedIn, Twitter, or Instagram are the channels that we use. And it's materials vetted by the scientific program holders. So the CDDF is always keen to hear more, to understand more about those who are attending, both on site and online. So our colleague Min will contact you and ask you to participate in a 5-minute interview. We would like to hear your thoughts about the workshop, about the CDDF. What can we do better? What is attracting you? So kindly talk to her and kindly accept her offer when she contacts you. Very important is how we engage here. So everyone should speak open and freely, and everyone should be free to use the information received, but not reveal the identity or the affiliation of the speaker, nor of any other participant may be revealed. So I heard this and that at the CDDF workshop is okay, but let me make something up. The representative of the Dutch Royal Court said this and this would not be okay. So very simple, and it includes social media. So please adhere to the Chatham House rule. That brings me to session 1. And my pleasure is to introduce my charming and very up-to-date co-chair. Katrin is also a member of the drug CDDF board. She has more than 20 years of experience in research and drug development, primarily oncology, immunology, and neurology. And I was very happy to share a few of these with you. And she has worked in an impressive range of leadership positions across different companies. And now, in the board and here at the program committee, you've done a lot to make this a successful workshop. So thank you, Katrin, and I turn the microphone over to you.
Thank you so much. And a lot of expectations. A lot of expectations now on me, Axel. Thank you so much.
I'm exactly on time.
Yeah. So actually, before we start, we have some thought-provoking presentations, but a lot of experts and technical views. We were thinking that it would be nice to actually level set us as an audience, and where do we start from for this workshop. So what we are going to do is so what we are going to do, we ask you a couple of questions where your head and what you're thinking currently is about the problem. So what we would like you to do, including us, is that we go on Slido, www.slido.com, and then you enter the # and the number you see here with 1576442 to join the multiple choice questions. So give everybody a bit of time to log on. Okay, then we have the first question for you. Do you think there is a need to optimize dose selection in oncology drug development? Give it a couple of more minutes. You see we have a lot of online participants as well. Already almost close to 70 votes. So I think this is a pretty clear, like is where our head as an audience is. We clearly see that there's a need to optimize dose selection in oncology drug development. So there's a lot of consensus here. Then we will go.
If I find it very interesting that there are 8 to 10% who say maybe, and I would hope that we hear more from you, because I think some people have doubts and questions, and it would be really good to hear from them.
Yeah. Okay, yeah. So 8% are around 10 people, right? So hopefully we will get to hear from them. Okay, perfect. So then we go to the next question. Which is, what do you see as the biggest challenge with regard to optimizing dose selection in oncology drug development? So what do you think are the highest hurdles here? Is it methodologies? Is it, change of habits? Is it number of patients needed to do so, feasibility? One word, please.
Answers coming up.
Yeah. One popping up immediately, cost and time. So we have about 60 votes. It's interesting. Time and cost came up very quickly, but are also seen by the majority of participants as the major challenges to implement actually new dosing strategies for early drug development. Yeah, I think this is something that will be addressed by several speakers as well. Methodology also. And then last but not least, our last question is actually, if we have these challenges, like time and cost, is it actually worth doing so? And worth doing so is, what do you actually expect as a benefit if we do so? So can I please ask you to also, in one word, describe what you see as a benefit? If we lose time, if we have more costs, what would be the benefit to patients? Okay, so I think also a very clear vote. The emphasis of the benefit is really on safety and tolerability, but also very clear about maybe improved efficacy or better benefit risk for patients. Also very clear. Unfortunately, in the first session, we do not have our patient speaker because he has a health problem, but we had lots of discussions with patient representatives in preparation of the workshop, and we'll give some feedback during the panel discussion. But I think safety, efficiency, tolerability, improved benefit risk is exactly what we heard from the patients as well. With that, I would like to turn over to our first keynote speaker with some thought-provoking presentation. Mark Reiten. He's actually professor at UChicago Medicine. He is the director of the Center of Personalized Therapeutics. He is an expert in early drug development and the use of investigational therapeutics in oncology. Has over 500 publications, is a key expert in phase 1 clinical trials for cancer drugs. And he's also, of course, an oncologist and graduated with an MD from Yale University. So it's my pleasure to welcome Mark. We have recorded his presentation because he is obviously in Chicago at the moment and for the time difference, but he will join us for the panel discussion. With that, I would like to switch to his presentation. Thank you.
Good morning, good afternoon. I'm currently in Chicago, and I very much appreciate and am honored by the invitation to provide the keynote lecture in regard to this important meeting. And the title of my lecture is The Need for Dose Optimization in Early Drug Development. So I want to start by mentioning that I do have some relevant interest to declare. I've not provided a laundry list of companies, but importantly, I'm treasurer and co-founder of a U.S. nonprofit corporation called the Optimal Cancer Care Alliance. And I also serve as a patent litigation consultant and recently as an expert witness on behalf of multiple generic companies regarding optimal dosing of anti-cancer drugs. So I want to emphasize that our lack of dose optimization has resulted in what I feel are unnecessary medical and financial toxicities, a two-headed snake. And I think we're all familiar with the side effects of the drugs that oncologists prescribe. Patients are all too familiar. And while there are some toxicities that are unavoidable, there are others that can be ameliorated or possibly even eliminated if we optimize the dose. And there's also opportunities to reduce costs, and certainly if excessive doses result in hospitalizations or need for additional medications to treat those toxicities, that alone will reduce costs. And finally, I will discuss during this talk the opportunity to reduce costs of those drugs that are approved and labeled at an excessive dose. All right, so I want to start with an example of ibrutinib. Ibrutinib, the first BTK inhibitor, a highly targeted drug, an irreversible inhibitor of BTK binding to a cysteine residue. And while there was some attempt to get the dose right, at the end of the day, this dose is excessive. And because of this excessive dose, there are now other BTK inhibitors that are approved and marketed and that are preferred by many, if not most, hematologists. This is from a paper in Blood in 2017. And I believe these data demonstrate that the lack of dose optimization of ibrutinib results in avoidable treatment interruptions and reduced efficacy. A quote from the paper is that 79 patients had dose holds for adverse events, 73 of whom restarted therapy at 420 milligrams, consistent with the prescribing information, and 5 patients restarted at a lower dose. 1 did not restart therapy. And the graph, however, is trying to convey a different story. It's trying to convey that dose intensity is important, that more is better. Actually, what this shows is that dose interruption is bad. So it's not about the dose intensity, it's about dose continuity. And the high dose results in dose interruptions. And I believe that that's the root cause of this signal, that dose interruption results in reduced efficacy. So historical oncology drug dosing paradigms are all based on the inaccurate assumption that more is better. The standard approach has been to determine the maximal acceptable starting dose for a population of patients, reduce doses in individual patients based on adverse events, and optimize dose intensity rather than therapeutic index or even efficacy. And as illustrated by the ibrutinib example, if efficacy is impacted by dose interruptions for adverse events, then less is actually more. It's not just less toxic, it's more efficacious. The world of oncology has also been fascinated by the notion of precision. We've seen this for decades in our approach to dosing, trying to precisely determine the patient's body surface area and precisely determine the dose for each patient, with some experts recommending therapeutic drug monitoring for modern drugs to get the dose absolutely right. The National Cancer Institute continuously talks about precision medicine. Yet when you look up the definition of precision, and I apologize, this is a US definition. I'm presuming that the same definition applies throughout Europe as well. When precision is used as an adjective, as in precision medicine, it's supposed to mean very exact and accurate. And I disagree that when we use the word precision in oncology, that always implies accuracy. In fact, let me show you an example where it does not. This is a news article published in the Irish Examiner in 2018 on precision surgery, robotic surgery. The epitome of precision, right? Absolutely cutting at the precisely best site. 2 years later, the same newspaper reported that surgeons operate on the wrong body part 63 times in Irish hospitals, presumably using their precision surgery instruments. It should be clear, precision doesn't mean accuracy. You also see this, unfortunately, in our drug labeling. This is the 2015 version of the US FDA-approved labeling for Xeloda, which I will argue is precise but not accurate. This is a drug for which many phase 2 trials were conducted at a dose of 2,510 milligrams per meter squared per day. And the prescribing information tells you exactly how many 150 and 500 milligram tablets to take to achieve the label dose, here rounded, you might say, to 2,500 milligrams per meter squared per day in 2 divided doses. Clearly an attempt towards precision. This label was recently revised in the United States in December of 2022. And I would characterize the revised label as accurate, but not precise. The recommended dosage of Xeloda is 1,000 milligrams per meter squared or 1,250 milligrams per meter squared, or presumably anywhere in between. And then the last sentence states, individualize the dose and dosing schedule of Xeloda based on patient risk factors and adverse reactions. So this is accurate, but not precise. And unfortunately, we have sacrificed accuracy in the search for precision. So the potential benefits of using an accurate lower dose are reduction in the frequency and or severity of adverse events, improvement in efficacy, reduced treatment interruption due to adverse events, better patient adherence, not to mention reduction in cost. Certainly, reduction in the indirect cost of adverse events. And for a drug that is marketed and priced, it can and often does reduce the direct cost of drugs. These are not new concepts. In 1994, the International Conference on Harmonization, a global tripartite regulatory group comprised of Europe, Japan, and the United States at the time, published this guideline on dose response information to support drug registration, the E4 guideline. This was adopted globally over the subsequent years. And as a reminder as to how long ago 1994 was, this guideline was issued prior to Sweden voting in a referendum to join the EU. And if you were using the internet then, you were using Netscape Navigator. Now let's look at what this ancient guideline says. Dose response assessment should be an integral part of drug development. Completely black and white, the word integral. We use the word integral a lot in oncology, talking about integral biomarkers. This is fundamental to drug assessment, drug development. And we now finally in the United States have specific guidance in oncology, albeit a draft guidance, that was released in January of 2023. And this is the written document that encapsulates the concepts of FDA's Project Optimus and an effort to require dose optimization prior to drug approval. I think you'll be hearing more about this project and this document through the course of the meeting. And credit for this document goes primarily to Murat Shah and Stacy Short. And among others, this document cites E4. Now, there has occasionally been an effort to optimize dosing of investigational oncology drugs prior to registration. This is the best example shown here, temsirolimus. And this is the randomized dose-ranging phase 2 that was published in JCO in 2004, randomizing patients to 3 doses of temsirolimus, 25, 75, and 250 milligrams once a week. And as you can see from the slide, there was absolutely no evidence of a dose-response relationship. Thus, the registration trial used a dose of 25, and this became the approved dose. Here is another phase 2 trial in kidney cancer, like the temsirolimus one, of the drug nivolumab. This was a well-done randomized phase 2 trial ranging from 0.3 to 10 milligrams per kilogram every 3 weeks. As you can see, there is no relationship of dose to efficacy, dose to overall survival. This was not utilized in any subsequent studies or in labeling. The labeled dose, the initial labeling was 3 milligrams per kilogram every 2 weeks, a 15-fold higher dose than was suggested by this study. And further evidence that initial study was valid is evident from this study published in JCO just last year using low-dose nivolumab, 20 milligrams every 3 weeks for advanced head and neck cancer, chemotherapy alone versus chemotherapy plus nivolumab. A large improvement in survival, no evidence of toxicity. All right, so coming back to ibrutinib, what we see here is the pharmacometric memo, and the clinical pharmacology team leader was Julie Bullock, who is now at Certara, regarding ibrutinib. And this goes back to 2013, where FDA tried to ask the question as to what the right dose was of ibrutinib in CLL, and specifically addressed the question, is the proposed dose of 420 milligrams daily appropriate? And the FDA conclusion was no. The proposed dose is 2 to 4-fold higher than the lowest dose that resulted in maximum BTK occupancy and maximum clinical response, and told the company the sponsor should thus consider exploring lower doses in future development programs. A very prescient comment. So we need to refine and redefine our phase 1-2 strategy. The primary objective of phase 1 needs to be to determine a range of doses for a randomized dose-ranging phase 2 study, as exemplified by temsirolimus, and this is all spelled out in the new FDA draft guidance. The upper bound will clearly be constrained by toxicity. The lower bound should ideally be identified based on tumor-related biomarkers. These are not biopsy biomarkers of markers of unknown clinical significance and unknown analytical variability and unknown inter-individual variability, but biomarkers that we understand: radiographic tumor size, perhaps circulating DNA, perhaps serum biomarkers. The optimal dose needs to be defined on the basis of randomized evidence and a global assessment of dose, exposure, efficacy, toxicity relationships, with a focus on accuracy rather than precision. In other words, let's get it about right, not 3-fold or 15-fold higher than where we need to be. And specific methodology was also laid out in this effort, and I had the privilege of participating in this effort from the MDICT that was led by Leslie Seymour, and this was published in Annals of Oncology last year, Oncology Phase 1 Trial Design and Conduct, and it illustrates the approach, dose escalation cohorts, and eventually select 2 or more dose levels. I personally favor 3 for a randomized dose-ranging phase 2 trial to provide evidence of the dose-response relationship. If it's flat, there's no reason to use a higher dose. If there is a dose-response relationship, then the highest dose or an intermediate dose is going to be more appropriate if the toxicity is acceptable. I will also add that in the United States, the US Senate Appropriations Committee, in its appropriations for fiscal year 23, specifically for FDA, included a statement with the bill that was enacted asking the FDA to get involved in post-marketing dose optimization studies. And just to blow this up, the committee is concerned about the escalating costs, especially cancer drugs and biologics. This goes on to mention Project Optimus and acknowledge the work that was done there, and states the committee strongly encourages FDA to organize clinical trials in collaboration with academic medical centers and other federal agencies of marketed cancer drugs and biologics to assess whether dosing and frequency adjustments may decrease the cost of care and or toxicities of treatment without compromising efficacy. We also recently published a paper in JCO, first author is Garth Strobin from the University of Michigan, basically outlining how this can be done. I would encourage you to take a look at the paper, but it basically suggests that the US government can conduct such trials at no cost. It's basically a negative cost because of the high cost of drugs, which would be reduced in half the patients. And this is similar to an approach that has been piloted in Europe, described in this paper from the Netherlands by Van Am and Nijhoff, published in Annals of Oncology. The senior author is Gabe Sonke. I will add that the US FDA commissioner has also recently criticized insurers for doing too little on drug research, and our strategy offers an opportunity for private insurers to get involved in clinical trials. So in summary, toxicity-based dosing regimens are inappropriate for most modern oncology drugs. Precise dosing of most oncology drugs is unimportant. Randomized dose ranging trials with reproducible biomarker endpoints such as tumor size are required to determine the optimal dose, and post-marketing dose optimization studies could be funded by payers since such trials have no or even a negative net cost. Thank you for your attention, and I hope we stop talking about both maximally tolerated dose and BSA-based dosing. Thank you.
So, thank you so much. Mark, are you online now?
I am. Can you hear me?
Yeah, perfect. Thank you so much for your wonderful presentation. Very, very thoughtful, and really pleasure to listen also at the history of, 1994 already being raised as a major topic, dose selection in drug development. Since you are online and we have a bit of time, we were hoping that you also could answer a couple of questions, maybe immediate questions. So if somebody in the room or online has a question to Mark, please feel free. We have about 5 minutes to grill Mark on this topic.
Please just come to the microphones here and identify yourself and get the discussion started. While people are considering this, I may start. So thank you, Mark, for a great keynote lecture. For me, while I completely see from my practical experience and from everything you've said, I see the need to test a different range of doses. It doesn't seem that easy to me. So it's easy to define the higher range because that is likely to be the MTD or below that. And but then also in the example that you've shown from the publications, how do I select the other dose or the other doses? So is it the lowest dose at which some activity has been seen? But if I compare these two doses, quite likely the best dose is not either of them, but in the middle of it, if there is a dose-response reaction. And there are also examples of bell-shaped curves in terms of dose-response reactions. So can you elaborate a little more on your thoughts about how to do that, how many dose levels, and how to find them?
No, I think that's a great question, and I agree that the methodology is key here. So I think we have to first of all take ourselves out of the resist box and look for a reproducible anti-tumor effect, and so and we can't be so concerned about rapidly getting to the MTD. What we really need to do is once we begin to see evidence of an anti-tumor effect. And I'm talking, a clear regression of the tumor, whether it's, a 15% decrease, a 20% decrease, a 25% decrease, or a 35% decrease we're talking shades of gray, not black and white. And once we have that, we have evidence of an anti-tumor effect. And that's what we should be looking for in phase 1. What's the lowest dose with an anti-tumor effect, not what's the highest tolerable dose. And in order to find that lowest dose with an anti-tumor effect, we have to be smart about the patients we're enrolling, because we only enroll patients who have no chance of benefit, we will not find that anti-tumor effect. And so that's the way we need to think about this. Let's find that lowest dose. Once we find an active dose, it actually gets quite easy from there. And because then it's just statistics. I personally think the best endpoint for determining whether there's a dose response is the time to when the tumor grows as based on modeling. I'm not talking about PFS. I'm talking about a metric that was developed by Lawrence Claret when he was unfortunately deceased, but it was at Farsight in collaboration with Renee Bruno from Genentech. And they have demonstrated that the model time to tumor growth using advanced pharmacometric software program called non-MEM actually is very predictive of survival in a randomized trial. You can see across 2 arms a difference in TTG that's likely to translate into a survival difference. And so we can apply the same concept to dose response, look for a relationship between dose and exposure and time to tumor growth. And I believe this can be done with sample sizes along the lines of the temsirolimus and nivolumab studies I showed you, in the range of 35 to 60 patients per arm. And just as an example, when Amgen developed sotorasib, they enrolled 120 patients at the wrong dose. And so it's not hard to find 120 patients if you have an exciting drug. It would have been a much more robust program had they used the strategy I'm suggesting, and I think we would by now have a clear answer that less is better.
Thank you.
So we have a, thank
Can
you.
I
We
answer?
have, yeah, please.
Hi, Mark.
Oh, yeah.
Yeah, perfect, exactly.
So you can hear me. You've been crusading the world against the use of BSA. You are recommending the use of food effect studies early on. You're now recommending, and I fully agree, that we should also look at the minimal effective dose to start a flow of studies. So in your experience now, what would be the optimal flow of events? What do you do first? Identify the minimal effective dose, identify whatever dose, but not using specifications like BSA. Food effect, what should we approach first? Because there is a need, there's a major need, particularly in small companies, to understand what's the flow of events that I should best follow.
No, that's a great question. Look, a lot of food effect can be predicted based on preclinical studies. Not completely, but just from the chemistry of the drug, you can have a pretty good idea as to what, solubility and permeability of drugs are the major determinants of food effect. So there's a lot you can predict if you think about it, and you basically, and there's nothing wrong with getting the food effect wrong early on, as long as you fix it. And so you take your best guess on food effect, and that's how you start your trial. And it's not a problem to bridge the food effect later in parallel to a randomized dose ranging phase 2. That's just a simple PK question. But I would focus on, studying a group of patients in phase 1 that have a reasonable chance of benefiting from the drug if the drug is effective, and therefore be searching for that minimal effective dose. I think that of course also tells you that the dose is active, the minimally active dose, which is really what companies are looking for when they start their phase 1 anyway, particularly if it's a novel target. Is there any evidence of anti-tumor effect? And so you'll actually get that efficacy signal early, which for a small company then allows them to go raise capital. So it actually is much better than the traditional route where you just put on a bunch of patients, find toxicity, and say, well, we finished phase 1, now we're going to go do a phase 2, and then the phase 2 is negative, and investors get pissed off.
And how about using exposure? I mean, the intra-patient variation can be big, but the inter-patient variation is even bigger usually. Still, we're using now a fixed dose in all patients. If your pharmacokinetics suggest that there is large inter-patient variation, would you prefer going for an exposure-related dosing or for a dose?
Well, I don't think there's going to be a need for TDM if we're using accurate doses of drugs that are novel and have a good therapeutic index because they're relatively targeted drugs. But I think from the standpoint of understanding dose-response relationships, exposure response is more powerful. But with
Okay, well, it's time to move on.
Yeah.
Okay.
So we will have Mark in the panel discussion as well, so we can finish then. And also online, we have a couple of questions which we will note down for the panel discussion, so to make sure these are being also addressed from the online audience. So.
Yeah. Thank you. And thank you for sending the question. Please keep on. We are monitoring this, and we will try to bundle the questions in the best possible way in the panel discussion. Next speaker is Elena Garalda. She is a researcher and clinical investigator in Barcelona, Vallhebron, and there she leads the early drug development unit and is the director of the phase 1 unit there. She has been in this field quite a lot. She has published there. So I think we will get a very good perspective from the clinic from Elena, and we are looking forward to hear your talk.
Good morning. It's really a pleasure for me to be here today and be discussing about Project Optimus, how will this affect clinical trials and drug development, and speak about my clinical trials perspective. These are my conflict of interests. So in the talk today, we will be speaking about what the goal is when we're speaking about optimizing dosage. What are the problems that we are facing, at least some of them? Why or what's the rationale of using the optimal biological dose instead of the recommended phase 2 dose based on the maximum tolerated dose? Potential clinical trial design opportunities, and then finally my take-home messages. So let's start by the goal. So in reality, all of us who are dedicated to drug development and to doing phase 1, what we really want to do is to best characterize the optimal biological dose that is going to be efficacious, that is going to be less toxic, and that is going to represent the most convenient schedule and dose in the early drug development. And we want to do this, of course, in order to define this for further studies. We want to decrease the period of drug approval without taking any wrong decisions. We want to make good drugs reach to our patients faster. And we want to benefit the wider population, once again, as soon as possible. And I think it's clear, and in drug development during the last 10 years, we've done a lot about patient selection, but we have not considered so much dosage. And I think this project is really important because it's putting dosage once again as a key element in the success of a drug to move forward. I think one of the questions that remains is really, does one size fit all? When we're talking about the specific drugs, will one drug development model fit all the drugs? And this is something that I think it will be great discussing later. So what are the problems that we are facing right now as phase 1 and that needs to be addressed when we are thinking about new clinical trial designs? Well, it's a list of some of the problems, because this list can really be much more extensive, and why designing these trials, doing drug development as we're doing has its challenges. One is toxicities. Well, we now know that with the new types of drugs that there is, we're going to have different patterns of toxicities. It's not going to be the same pattern of toxicities that we had with chemotherapy, where they were all very acute. Here we're going to have acute, but we're also going to have late onset and also chronic toxicities, particularly with the molecularly targeted agents. We know that there are different variables that are going to be affecting dosage. For example, the tumor setting. It could be different in the early stage versus late stage, the tolerability of the drug. And this could depend, for example, on the difference of tumor burden. It could depend of the different tumor types. For example, the tolerance of regorafenib in GIST tumors versus of metastatic colorectal cancer patients. It's different. It could even depend of different molecular profiles. We know that we need different doses of imatinib in GIST that have exon 9 mutations versus exon 11 KIT mutations. So it really, that has a lot of variability, the dose that is going to be correct. It also depends on the type of drug that we are developing. It's different if we are developing an antibody than a kinase inhibitor, a small molecule. Or even if it's a specific inhibitor, like versus one mutated gene versus more multiple targets in a more promiscuous way, the toxicity profile is going to be different and the doses is going to be different. Also, is there's correlation or no correlation between dose and PK/PD effects? And finally, that the definition of the optimal biological dose based on efficacy and toxicity, we're also needing to counterpart other aspects such as conveniency based on efficacy toxicity, but also on convenience. And as I've mentioned, we are now developing multiple types of drugs, not only chemotherapy, and they have a lot of particularities. And in this slide, you can see some of the differences that are going to make us different how we develop these drugs. We're going to have drugs that have a really very limited off-target effects, such as mutant and isoform selective agents versus multi-kinase inhibitors that are going to have high off-target toxicities. We're going to have molecules where the therapeutical window is very narrow, and others where it's very wide, or even it's unclear if we can really reach a maximum tolerated dose. And we're going to have treatments where the target population is highly selected, versus other where we don't even know how to select drugs. So when we are talking about drug development, we have really very different clinical scenarios. So what have we done until now? The reality is that, as we've heard also in the first talk, what we were doing in phase 1 is really having very small, very fit patients that went into small cohorts and escalating based on toxicity. So we were really looking the maximum tolerated dose based on these toxicities, that mainly the DLT. So we didn't consider other safety, long-term toxicities, or treatment reducement. It was a very short observation period. And finally, we would select the maximum recommended dose based exclusively in toxicity based on these DLTs. This would be the recommended dose that we would move for a registrational study. So what is really Project Optimus and this new optimization strategy proposed by the FDA, Project Optimus? Well, here we are talking how before this registrational study, we really need to have a dose optimization study. And this will be discussed also later during the whole of this workshop. So we will need randomized information about different doses before really going to the registrational study. So we have a dose optimization process in the middle. The question is, of course, also, will this take more time? No? So how can we really improve dosage in the clinic? Well, it's clear for me that we need to evaluate safety beyond DLTs. The DLT concept comes from when we were developing chemotherapies. Now we have different types of drugs. It's clear that we need to go beyond them. We need to go beyond also considering only toxicity. We now have much more valuable data that also needs to take part of this decision. PK, PD, efficacy, safety, and tolerability as each step need to be considered. And of course, all the preclinical data too. We need to really be careful and have clear the characterization of the dosage and exposure relationships for efficacy and toxicities. We really need to understand how we increase dose, we increase exposure, and what this represents for efficacy and for toxicity. And ideally, what we want is to really identify a dosage range early and then further evaluate several doses, ideally in a randomized way. So what are the challenges? Well, the concept is really not new, and there have been several efforts from academics and from different stakeholders to talk about dose optimization from some years already. But it takes time really to change, and I think this is the first challenge, that we are really not used to doing it this way. So there are some perceptions that need to be overthrown in order to be able to perform this change. The first challenge that there is in the clinic is the maximum tolerated dose paradigm. We have in our heads that the higher the dose, the higher the efficacy. And this is throughout stakeholders. Sometimes when I am for example, informing our referrals, say, we are already in dose 4, saying we are already at higher doses. This means that the patient has a higher probability of success. So it's all stakeholders. Patients believe that the higher dose, the better. Physicians believe that the higher the dose, the better. Referrals believe that the higher the dose, the better. So this is something that needs to be done. And pharma companies also think that the higher the dose, the better. So we all need to be educated that this is not necessarily the case. Also, the informed consent is necessary. This needs to be reflected in the informed consent. The informed consent to the patient should really document clearly the reasoning for various dosing. And we need to have trial design considerations such as crossovers and interim analysis that protects patients from doing this. We also have to combat the perception that this is too time consuming. We have to understand really that ill-optimized dose can really on the long term impact in a negative way the ability to document the true benefit of a drug. If we really believe that the communication with regulatory agencies will be quick and fast, in the end, the process will be efficient. And in the end, this comprehensive dose selection early in the beginning will really allow for a more rapid development of new therapies, for example, combinations, modifications, post-approval, new formulations, etc. So the perception that is too time consuming needs also to change. So in that regard, I did want to finish explaining how we did convene the MDICT workshop and publish these guidelines in Annals of Oncology with a proposed new model to perform phase 1. And this has also been spoken in the first talk briefly. And this is the guidelines that were published in the Annals of Oncology a couple of months. And here you can see the typical dose escalation where we are reaching the maximum tolerated dose. But really from the very beginning, the recommendation is we really should identify this recommended dosage range. So what doses we're going to be studying further in this dosage ranging study that will go from the minimal reproducibly active dose where we're going to see, for example, tumor shrinkage, circulating tumor DNA, etc., up to maximum tolerated dose. So we do our classical dose escalation until maximum tolerated dose, but we are also going to be having these dose ranging studies. And the reality is that there's many designs that can fill in the phase 1 where we can have having more information about these doses. For example, some is actually backfilling in selected dose. So once again, you have selected these dose ranging doses that you're going to be selecting. You can backfill to selected doses in order to have more information. And once again, this information is based on the totality of the data. These are the doses that you are going to move forward for backfilling. And even you can include randomizations from the phase 1 trial, and the randomizations could occur at different places. It could occur, for example, once you're including a patient in the trial, you could randomize the patient to continue escalating versus randomization to the backfilling cohort. Or you could include the randomizations already in the expansions. Whatever the situation or the randomization, the endpoint is you're going to have more information at different doses based is going to allow you to optimize this dose ranging interval in order to get more information for selecting the optimal dose. So what are my take-home messages and as a clinician? Well, I think regulators and all stakeholders are challenging the paradigm that assume that the more is better. This is challenging, but I think it's essential. It's the first paradigm that we need to break. Patient-focused drug development is not only an idea. It will happen, and the early drug development field is not an exception. Actually, it is the piece to start. So this needs to start from the phase 1 trials, and it's very important that patients and patients' advocacies also speak about how they feel about these new concepts. Instead of choosing a single recommended phase 2 dose, we will be asked to provide a range of doses to be further explored in more advanced phases. Toxicity, PK parameters, and clinical efficacy will be used, but also pharmacodynamic markers, patient-reported outcomes, PKP modeling, and even costs will take part of the consideration. So we will be doing a more holistic integration of all the data in order to recommend this range of doses. And in the end, I think one of the challenges that I think it will be good to discuss in the panel is whether this context should apply to all types of drugs or this is more specific of certain types of drugs. I think flexibility is really going to be key, flexibility with the regulators, flexibility in the clinics with the academics in order to understand when this concept is particularly useful and when it's going to be more important. And with that, I want to really thank you all for your attention. I will be very happy to take any questions during the panel discussions. Thank you very much.
Thank you very much. Hello, nice to see you. Thanks for joining us. We have a ton of questions online, so maybe we start with that before we go into the questions. Because we have so many questions, would be great if you could vote on them. You could just touch the thumbs up symbol, and the questions will, or that will give a signal to us that the question has more interest than another one. So I need to take my glasses off for that. But I think one of the question was, how do we optimize exposure when optimal exposure varies for individual cancer cells within a patient, so over time between patients, between tumor types, etc. Do you have an answer to that?
Well, I think Mark started speaking to this before he got frozen out, so we can discuss it in the panel. But the reality is with the new drugs, when we have also very efficacious drugs, also the exposure is going to be very, very relevant and very consistent in a way. So I think probably, and I think it's good if Mark finishes what he was going to mention, but probably if we have a clear understanding of how this exposure dose exposure and then exposure toxicity or exposure efficacy goes, we can take our general approach that it will not need to be individualized to each of the patients.
So a question that is really trending is the sample sizes. So can you say something about what are the necessary sample sizes? So the FDA guidance says, well, they don't need to be fully powered, which in a way is reassuring, but it doesn't really say what is expected here. So that was also one of the questions I wanted to ask basically each of the presenters.
So the quick answer is I don't know. I mean, I think it's clear, and this is also when we are designing the trials and when pharma is asking what is the number, I don't think it's true that the guidance is clear on the number. And this is probably because it will depend on the drugs. I mean, I think what is important is the concept, but as I was mentioning in the presentation, probably it's going to be different depending on the drug that we're going to be using. So perhaps in depending on the type of drug, there's a clear difference between the doses, and there's doses that have different PK profiles, different efficacy profiles, and the numbers that you're going to require is much less. And perhaps there's trials or drugs that there isn't such a difference, and you're talking more of an optimization of toxicity or cost. And I think perhaps this is going to be more subtle to see. So I don't think there's a clear, or at least in my opinion, a clear number. Is if concept-wise you are able to determine these dosages to perform later. But I don't know, Mark, I think you are also connected. I don't know if you have a more clear number to answer.
Well, remember, really the way to think about it is once you defined a minimally effective dose, you're looking for evidence that there is a higher dose that is superior. So that's what you're looking for. You don't have to prove that. But if you can't find any evidence that a higher dose is superior, there's no reason whatsoever to use that higher dose. And so that's one way to think about it. The optimal sample size, nobody can know at this point in time. You can only know that afterwards. There may be some work that could be done based on resampling completed trials, after the fact, or perhaps even resampling the trials for, and again, we've got such a limited portfolio of trials to even study. As I mentioned, there's the temsirolimus and the nivolumab trials, neither of which show any relationship between dose and efficacy. And there was also a trial of paclitaxel years ago conducted by the former cancer and leukemia group B. And so we have a very limited group of trials, but I think figuring about 40 to 50 patients per arm in a 3-dose study, in other words, something in the range of 100 to 120 patients is a very reasonable number. It's the kind of numbers companies are used to thinking about anyway if they want an accelerated approval using response rate as an endpoint. So think about it this way: if you put 40 patients on each of 3 doses, and you see evidence of a dose-response relationship, okay, well, that's easy. You've got evidence of a dose-response relationship, and you have evidence for the higher dose being more effective. If you also see evidence of activity but no dose-response relationship, well, you've got 120 patients treated at a dose of the lowest dose or higher. You also have evidence of efficacy, and the only thing you don't have is a very large safety, safety database. But you're going to have to do a post-marketing study anyway. So I don't see any downside to really doing a proper dose-ranging trial, which can, in and of itself, result in accelerated approval.
Thank you. We have to move on, but don't forget there will be a panel discussion. Please keep voting on the questions.
So I have the pleasure to introduce our next speaker, Philip Josefsson from the Medical Products Agency in Sweden. Philip is a clinical assessor and currently the alternate CHMP member for Sweden. You are a medical doctor, and you have been involved as a rapporteur and co-rapporteur in multiple assessments of new oncology drugs in the European Union. So you will give us the regulator's perspective, so very much looking forward to your talk.
Thank you very much, and thanks for inviting me to speak here. So I'll give the regulatory perspective, or rather the European Union regulatory perspective, I should clarify. And whatever I say, even though this might be my own opinions eventually, rather than the position of the institutions that I represent. So starting out with, what are the sort of requirements for licensure in the first place? So I'm I'm quoting the law here first, saying that the marketing authorization shall be refused if A, the medicinal product is harmful in normal conditions of use, or if its therapeutic efficacy is lacking or is insufficiently substantiated. So that's expressed sort of in the negative, and correspondingly, one may understand this, that if the benefit-risk balance may be deemed positive for the proposed use, the marketing authorization should be granted. So where does that place optimizing the dosing regimen and essentially the requirements of the ICH-E4 document that was cited? It remains that there's no formal requirement to establish the optimal, whatever that may mean, dosing regimen. We don't deny approvals because another unstudied posology might have had a better benefit risk than the one studied and proposed by the sponsor. Moreover, as has been sort of touched upon here, a truly optimal, scientifically optimal characterization of the exposure-response relation for a medicinal drug may require the exposure of patients to doses that are a priori likely to be suboptimal. And of course, in some treatment settings, this may be a problem. And it remains that as regulators approving clinical trial applications, study subject safety is the primary concern that we look at. Obviously, we want the studies to be scientifically, valid and competently designed, but ultimately study subject safety is what we're there to sort of guarantee in some sense. And as I said, under some circumstances, there may be a tension between this and the complete understanding of exposure response. So what about post-authorization? There may be room for improvement, we know. So post-authorization commitments to study alternative dosing regimens after EU approvals are not the rule, but they do occur, and occasionally they've involved the randomized comparison of the approved dose and the lower dose. This is reviewed, the European evaluations of oncology drugs with respect to dose exposure response has been reviewed by Maljapard et al. In 2021, a very nice paper that sort of describes this in more detail. We all know that in some cases, medical practitioners have investigated and or adopted lower doses than those originally approved according to the SMPC. And sometimes these have become simply generally recommended dosings. And such lower doses have been accepted by us for use in pivotal trials for new agents, for instance, as backbone or as a potential comparator, if they have been generally used clinically. And I think bortezomib and capecitabine would be two such examples. In some cases, insufficient evidence for such dosing regimens accepted by the clinical community have prevented their regulatory approval. And I think underlying this lies a very important question. If a drug is approved with a certain dosing regimen and there's concern that this is excessive, what kind of evidence should we require in order to say that, say, a lower dose has an equally good benefit risk and can substitute for the higher dose? Do we need randomized controlled non-inferiority trials with, a tight M2, or could this be done in different ways? What is the role of exposure response? What might be the role of real-world evidence? I think that's a very important question that I don't have the answers to at this point, but in any other way than what the practice that we've had over time. So having said this does obviously not mean that we're uninterested in dose exposure response and dose selection. And there's a guidance document from the CHMP, the anti-cancer guideline, which describes this to some extent, the European regulatory thinking about this. This distinguishes between cytotoxic and non-cytotoxic compounds. for cytotoxic compounds, the basic assumption governing the design of single-agent dose and scheduled final trials is that toxicity is an acceptable endpoint. So the main objective would then be to define dose-limiting toxicities and the dose to bring forward into further trials. MTD, DLT, and recommended phase 2 dose should be identified. That's obviously the old-fashioned paradigm that the previous speakers have alluded to. The non-cytotoxic compounds, on the other hand, are described as a very heterogeneous group of compounds. In contrast to cytotoxic chemotherapy, these compounds are typically administered continuously, and the toxicity profile tends to differ so that those limiting toxicities may occur first after multiple cycles of therapy. And this scenario was obviously also alluded to by, I think, both of the previous speakers. And it's conceded that this may require alternative strategies with regards to the definition of those limiting toxicities and the MTD. With respect to how this dose-finding procedure is envisioned, it's stated that the dose-finding strategy should not only focus on safety endpoint, but also on determining an optimal dose. Very much the same kind of language that we've been hearing here and that we have in the FDA Optimus document. So this would be the dose at which optimal biological response, according to a predefined effect marker, is achieved. At this dose, it's presumed that giving a higher dose does not further improve outcomes. And I guess the key sentence with respect to how this is envisioned is examples include escalating doses until a target-mediated biological pathway is optimally altered, or escalating doses until a target becomes saturated with the drug, while minimizing the dose required to achieve this maximum pharmacodynamic effect, thereby aiming to minimize toxicity. And I think everyone could probably agree on this. The question is, what does this entail in practice? What investigations, and how do we design those? And I guess that's the key thing we're here to discuss. So now I'm going a bit off script compared to what's in our guidance. The previous speaker referred to the differing concerns with different kind of compounds. And for protein kinase inhibitors, in the general case, with increasing exposure, such agents will become less selective with broader receptor promiscuity and more off-target receptor effect. And I'm saying receptor here as opposed to off-target tissue effects, which we'll discuss with antibodies. Correspondingly, with lower doses than anything, I guess, there may be less off-target receptor effect depending on how the exposure-response curves look for the different the drug's engagement with different targets. So we've already seen a few suggestions on how a rational phase 2 randomized dose-ranging trial might look, and I think what I'm saying here is sort of in the same line as what we've heard previously. This might include a dose defined by target saturation or as the maximally tolerated dose, and compare that with a lower dose by some modicum, whatever would seem reasonable based on initial PK/PD. The study objective would be to determine if a lower dose might be similarly active or exert similar PD effects while showing better tolerability. And the question of sample size as well as the duration of such a study is obviously key issues. And as stated previously, there would be no standard answer to this. This would be decided by the precision in the PD markers and the safety endpoints required, whatever is actually the scientific hypotheses of this study, and what would be required in terms of precision in order to make an informed and clever decision on which dose to take forward. Now to intrapatient dose modifications for protein kinase inhibitors. It's already been noted that one dose is not going to fit all. The relation between dose and exposure will differ between patients, and the relation between exposure response might differ for several reasons, as well as the tolerability as a given exposure. So this is obviously well recognized, and most pivotal trials of protein kinase inhibitors investigated dosing regimen with a starting dose And part of that regimen is a dose reduction scheme. And for a variable and frequently significant proportion of patients in pivotal trials, the starting dose of a protein kinase inhibitor will not be tolerable, and dose reductions are necessary. So one may have a few reflections on this. Firstly, a high proportion of patients requiring dose reductions may not per se indicate that the dose selection was wrong, provided, of course, that most patients eventually do not need to discontinue therapy, which I guess is the whole sort of rationale for dose reduction schemes in the first place. However, once you do a study with one starting dose and dose reduction schemes, it's going to be very difficult to base a PK/PD model only on that study in order to tease out whether a lower dose starting dose might be good. And that is obviously because the ER will be confounded to some extent because those who require dose reductions will not be, random people. They will be people who either had high exposure at a given dose or else who did not tolerate the exposure they got, whereas others might have done so. So they're not ideal actually for as a basis for exposure response modeling. Monoclonal antibodies, a little bit of a different scenario, at least in my mind. For a conventional monoclonal antibody, not toxin-linked and not a T-cell engager, no off-target effects would be anticipated. Side effects would be anticipated because the effects are not limited to the tumor. Presumably, I guess dose finding should generally, in these cases, ensure that the selected dose yields exposure beyond Emax on the plateau of the exposure response curve for most or all patients. Notably, monoclonal antibodies tend to have target-mediated clearance, which may be variably important, and this may correlate with prognostic factors. A large tumor might give a larger target-mediated clearance, so large tumor burden. In such situations, randomized dose-ranging trials may again be required to inform an unbiased exposure-response model. And we've seen that the exposure-response model for antibodies have been of key importance for subsequent adaptation, for instance, of the dosing interval, as we've seen with the PD-1, PD-L1 inhibitors, or when bridging from an initially studied IV formulation to subcutaneous formulation. And I think antibody-drug conjugates are very interesting with respect to the discussions we're having, because obviously they would sort of fall into two different brackets at the same time to some extent at least. So I'll finish with a few. I don't pretend to have a lot of answers on how to do this best, but I'll finish with a few questions that I hope that we regulators can learn more from you all at this meeting. Firstly, what safety problems can and cannot be addressed by dose titration in individual patients based on dose reduction schemes, rather than optimized starting dose selection based on randomized dose comparative trials? Under what circumstances is a randomized dose comparative study as proposed by the previous speakers and by the Optimus document, under what circumstances is this needed or advisable? And what are appropriate target populations for such studies? And if a study is advisable, on the basis of what research questions is that, and what are the appropriate corresponding study designs and endpoints, including the relation of this to study size and study duration? If long-term toxicity concerns are key to the dose comparison, how are they realistically addressed prior to phase 3? And this sort of begs the question whether there are situations where the rational thing is actually having more than one dose in phase 3, because the study you're performing is going to be of a size or duration that would make it a potentially registrational trial anyway. And finally, I think it's a very interesting question. It sort of links up with all of this. How do these concerns and solutions on dose selection differ between kinase inhibitors, monoclonal antibodies, antibody-drug conjugates, and also perhaps T-cell engagers that we're starting to see now? So, thank you very much.
Thank you.
Thank you very much, Philippe. I think also your questions, maybe that's something we can discuss, also take up again in the panel discussions tomorrow when we have heard, about new modeling, new methodology. So I think it's a very, very nice way also to see tomorrow if there are some ways to address the questions you have here. I guess these are the questions most of us have in the room, and for which it will be difficult. But maybe with some of the discussions over the next 1.5 days or 1 day, we will find at least some recommendations, hopefully. Thank you so much for your talk. Very, very nice to get, getting the food for thought here, also what open questions we still have to address. We know that we want to change, but how is as you said, is the big issue here. And I wanted to open also for you a quick round of questions. Anybody in the room who has a question to Philippe before the panel discussion? The moment we don't have one coming through, I always look at the most recent ones. Yeah. And please state your name and affiliation.
Yeah. Hi, it's Jessica from Janssen. Thank you. So I think there was some thoughts earlier about randomized phase 3 registration trials, and I heard some ideas as well of the possibility of a 2-cohort, 2 different dose levels being serving as, can we model those to be one efficacious cohort against a control arm or something? Are these concepts that would go? Like you have 2 different dose levels and you kind of make the efficacy based on the whole cohort, but look at other kind of aspects when it comes to approval of a certain dose. Are these flexible mindsets that are approachable, you think?
If I understand you correctly, there's 2 scenarios. I mean, one thing is what's required for the initial dose to be registered. And what kind of, I guess, flexibility here means what kind of supportive evidence where statistics are, if statistics aren't sufficiently precise due to limited size, what is the role of PKPD and so forth. And I think that's obviously very important going forward. But I don't know how willing we are to sort of use PKPD to propose a different dose from that actually studied, for instance. I think that's a tricky question.
When you move to the next phase, like the phase 3 trials from the initial dose, that was the kind of transition.
But that would generally be the case, wouldn't it? You would need to integrate not only the clinical data, but also PKPD and perhaps translational medicine aspects, because you're not going to do, because if you don't do that, eventually your dose finding will be as large as your it could be your pivotal trial that's at some point. And then with respect to once the dose is approved and the idea is to optimize this perhaps with a lower similarly effective dose, I think that's a very difficult and very important question. What are our societal expectations of evidence of equivalence with respect to efficacy in such a situation? I think if other people would express their views on that would be very nice.
Thanks.
We have an online question, but I allow myself to tag on a second question to the online question. So the first question from the online audience is, are there any discussions between the EMA or the CHMP or the Oncology Working Group with the FDA on Project Optimus? What are your exchanges?
We've been exchanging on that, yes.
Okay. And then my question to you is, we have this more complex system in Europe, where actually you in a national agency see the early drug development because you see the clinical trial applications that's coming your way for clinical trial conduct in Sweden. But most probably as an assessor of a marketing authorization application, you see it once or twice during scientific advice, but then you are presented with the final data. So what is your approach at the Medical Products Agency at the moment when you give advice on clinical trial applications? Are you actively recommending to include an element of dose optimization when you see clinical trials coming your way?
I think that realistically what we do is we look at the proposal of the applicant and we see if this is, firstly we check that this is compatible with patient safety, and then we sort of in a more broad way want to ascertain that the sort of experiment that is proposed is scientifically viable. But I would not be telling the truth if I would say that we view each clinical trial applications within the framework of the entire development program. I think that's probably one of the challenges of the European system, that we're less able to do that, because the relation between those that approve clinical trial applications in different countries and the groupings that ultimately approve drugs are maybe, as you said, look a little bit different from what is the case in the United States.
Okay, thank you very much. I think with that we will move to the last presenter. Thank you, Philip. We'll see you at the panel discussion. Oh, please, sorry if, yeah, sorry, I didn't see you. Please, sorry, come back.
Well, I just had a follow-on question to your question, Katrin. So, from a sponsor perspective, we obviously look at global development programs. So with Project Optimus coming on board, as we design our phase 2 expansion cohorts or randomized cohorts to look for dose, that flexibility that you were talking about, I guess, in the European system of evaluating clinical trial applications at a national competent level, we need to see some level of greater flexibility in terms of stopping enrollment or expansion cohorts, especially with EU CTR coming on board to avoid substantial amendments. So how does that play into what's coming through from the FDA, but also where you guys sit at the MPA of managing such amendments?
I think that's a difficult question that I don't think I'm capable of answering at this point.
Yeah.
That's a good question.
Yeah, thank you.
Yeah.
Honest answer. Yeah, thank you. Okay, then.
Bring the remaining questions into the panel discussions.
Yeah.
So my pleasure is to introduce the last speaker of this session, our co-program committee member, Harald Weber, who is an Executive Medical Director of Early Clinical Development at CGIN, and in this position he has a lot of experience in these questions from an industry development perspective. And Harald, please, the floor is yours.
Yeah, thank you very much, Axel, for the very nice introduction. It's a great pleasure to be here and to provide some overview and some reflections on how the industry has reflected on these challenges of dose optimization so far. Of course, as a disclaimer, all the opinions and all my statements should be seen as my personal opinion and not of my employer, by CGIN or CGIN International. The objectives of the talk, I basically see two areas that I wanted to talk about. First, I wanted to reflect on the key challenges that we have seen so far, also in the phase 1 studies that we have been conducting so far, and the open questions that there are still existing when we go through FDA's Project Optimus draft guidance. And I wanted to also discuss some early considerations, and also maybe to feed the panel discussion later on, how this could be implemented in oncology early clinical drug development. So this is a sort of a repetition of what you have seen before, as Mark presented this so nicely. But I wanted to keep it in my talk, because I have to say I find it quite amazing that it has taken more than 20 years or even longer from the ICH E4 guidance on drug dose optimization, with many steps in between, mainly driven by the FDA, until we have finally arrived at the optimizing dose optimization for the guidance for industry as issued by the FDA. And it seems to me that change takes time to be adopted, especially in innovative changes take a lot of time to be adopted. So if we rethink how we conduct early clinical development in oncology today, I think the current situation is characterized by significant changes, as you all know. The number of approved anti-cancer medicines in Europe has more than doubled in only 5 years. And many of these agents are targeted agents, are new molecules, new mode of actions characterizing these new drugs. And we have, at the moment, according to clinicaltrials.gov, more than 6,000 active clinical trials investigating only novel immunotherapies. So there is clearly a very high need to look into those optimization efforts in more detail. Targeted therapies are often developed, as also mentioned by previous speakers, are very often developed for chronic use, for continuous use, until relapse occurs or progression of disease. And our phase 1 studies that we have been conducting so far, using the dose escalation scheme that you're all familiar with, we look at the MTD and the dose-limiting toxicity only, but those trials are completely insufficient to learn anything about long-term toxicities, chronic toxicities. So I think the goals from my point of view for dose optimization are basically to, on one hand, improve tolerability, by doing this by dosing, reducing the dose modifications and dose reductions, so that longer treatment duration is actually possible. To accelerate also patient access to new cancer medicines, and to avoid dose optimization in phase 3 or in a post-marketing setting. What are the dosing challenges in the era of targeted therapies? And I did some literature research here and found quite interesting data, very much in line with what Elena and Mark presented before. The first one is there was a publication several years ago that in more than 1,200 patients with metastatic breast cancer who have received the registered dose, the approved dose of an anti-cancer drug, 86% of these patients reported adverse drug reactions, so related adverse events, with 43% of those patients missing at least one dose, and 20% of those patients requiring a hospital admission. Obviously, this has also some impact on the costs of the healthcare system. The long-term use of a less tolerable dose in a situation where lower doses would be sufficient may also cause poorer compliance and lower efficacy. There was a publication several years ago by MD Anderson. They have looked into 24 phase 1 studies and found that a substantially higher dropout was seen due to toxicity, with no improvement in response over doses that were about half of the MTD. And in only 30% of more than 200 phase 1 trials reported objectively quantifiable or clinically gradable adverse events. So I think Mark has highlighted this before as well. We need to look also into not the grade 3 and the grade 4 adverse events or adverse drug reactions, but also get a better picture about the subjective adverse events, pain, fatigue. Axel, you mentioned this also in the introduction. Roughly 2/3 of all monotherapies that have been approved are actually approved at doses that are less than the MTD. But the good news is that many of the registered molecules or compounds, the dose is actually less than half of the MTD, 32% based on publication by Sachs et al. The dose slightly below the MTD in about 30% of the cases, and in 38% the dose is at the MTD or higher. Of course, this picture is not, has to be taken with a piece of salt, because it doesn't take into account the mode of action. Is it a chemotherapy, targeted agent, monoclonal antibody? So the examples of successful non-MTD development that I've found in the literature are basically modeling and simulation-based methods. So we will have a lot of more discussions about methodologies, taking also preclinical and early clinical aspects into account. biomarker-based dosing approaches, and these strategies have obviously, for at least about 60% of approved drugs, already been successfully implemented. So I'm sorry that this is a very small picture here, but I found this, and I think it was referenced before Malipart ESMO opened 2021, an overview of observed dose selection criteria for 60 agents in EPARS between 2015 and 2020. And on the left-hand side, what you find there is that the MTD was defined. The larger part in the middle, the MTD was actually never really determined. And on the right-hand side, those molecules or these approved drugs, the MTD needed to be reassessed later. What is also fascinating is that in some of these molecules that have undergone post-marketing or post-authorization measures, that some of these molecules were finally withdrawn from the market. So I think that the goal, the target, is essentially to move away from the MTD to something that, some call it the optimal biologic dose, others call it the minimal reproducible active dose, that we, based on the totality of the evidence, that we get a more robust understanding on the impact of different doses on efficacy and toxicity. And that includes chronic and persistent collection of low-grade adverse events. The target of these new molecules, the targets may be saturated far below the MTD. So there's less on-target, there may be less off-target toxicity as well. And I think it was mentioned also before, for the randomized comparison, I think the aim should be to identify the minimal biologically active dose and the highest tolerable dose, and maybe some dose levels in between, and then to rule out if there's a difference in the overall benefit-risk assessment to finally pick the most appropriate and most promising dose. But, and this is, I think, very important for all stakeholders, that requires more patience in early drug development, that increases the heterogeneity of the patient population that is to be included in phase 1 studies, and it, of course, requires more time. I wanted to say a word about heterogeneity of the study population, because FDA's guideline is actually asking for enrollment of an appropriately broad population to assess the impact of all covariates on PK and safety and efficacy. But with that broader population that's being enrolled into clinical studies at early phase, we automatically increase the heterogeneity of the study population as well. So what would be the impact on the data interpretation? On one hand, the advantage that I see is that the PK data that is being collected can sufficiently support the population PK analysis and dose exposure response analysis. However, it's not always consistent across different populations that we may enroll in the dose escalation or the dose optimization phase of our phase 1 studies. There are differences in tumor types, maybe due to additional signaling pathways. There are different mutations that need to be considered as well, which have an impact on drug affinity or the activity of the inhibition that we want to achieve. And it's, I think, important also to integrate early in our development strategy a proper analysis of pharmacodynamic and pharmacogenomic data to be considered. Now, the open question remains later for the panel discussion and then also for the sessions tomorrow: how do we balance the need for more data from a broader population on one side without diluting or confounding, instead of strengthening the interpretation of the data to derive at the right conclusion? There are other aspects that I wanted to highlight as well, which just keep adding to the overall complexity. Here in this case, in terms of study designs, as we have heard repeatedly now, the recommended design is a randomized parallel dose response trial. However, we may be forced to enlarge the dosing intervals between the different cohorts because we wanted to, and we have to collect more data to capture PD and long-term toxicities. Model-based systems, or from the biostatistical point of view, may need to be integrated later in order to be considered, but then more resources are required. And we wanted to ensure, on one hand, similar populations and interpretability of dose and exposure response analysis. However, that goes in contrast to the request or the guidance to enroll a broader patient population. So timing aspects and the prioritization of those comparisons have to be reconciled also with our plans to expand into different tumor types. And that is needed before the dose efficacy relationship can be fully explored in an adequately sensitive population in a registration-enabling study. So we need to use real-time PK data, again, with the implications on resources and capacity, and early validation of PD biomarkers to support development decisions. And we will certainly hear more about adaptive study designs, utilizing interim analysis to allow early stopping of one of these dosing cohorts or dosing arms in randomized studies. We have heard it also before already that the typical endpoints that we are looking at the moment, response rate, duration of response, may not be entirely adequate anymore. So we need to leverage advances in radiographic or ctDNA and blood biomarkers, do probably some sampling at multiple time points in order to have a lower variability and greater accessibility and acceptability than biopsies do. Because as we know in phase 1 studies in patients with advanced disease, it is usually very challenging for the patient to get a biopsy obtained for research purposes. And the validation of biomarkers have to be done much earlier than in the past, I believe. And that comes on top of the complexity that is already added by IVDR. So this has also implications on resources and costs. Some additional points to consider, just to highlight, including PROs, engagement with patients, a higher frequency and impact of symptomatic reactions, grade 1 and grade 2 adverse events, and what do we need to do in order to support subsequent indications and usages, considering non-clinical and clinical data, and a strong rationale to be built to support the dose selected finally for the registrational study. Speed was mentioned at the beginning as one of the key concerns or key areas of interest, because the overall duration of drug development, if we do all these or take all these complexities into account, will definitely affect the need for an increasingly rigorous dose characterization. Hopefully, if we do more at the beginning, that may be balanced by yielding more optimal dosing for registration studies. Now, it could have an impact also on fast-track approaches. When we do more at the beginning, that might be possible to streamline regulatory interactions, such as fast-track in the US or in the UK or PRIME in the EU, to overcome challenges of increased timelines. Another topic for discussion and consideration. And of course, when we talk about speed and time, we talk about costs. I think this is something that is, especially from the industry perspective, important to be considered, because these changes in clinical trial designs and additional data requirements, additional analysis, would definitely have an impact on funding, especially for smaller instead of smaller biotechs compared to large pharmaceutical companies that have a higher degree of flexibility to rapidly increase budgets. So in conclusion, I think the primary goal remains, as discussed by other speakers before, to support the paradigm shift, moving away from MTD and to use an approach that is allowing us to define optimal doses earlier in our clinical development strategies, and to do this by rigorously selecting dosing schedules that also help patients to gain an increased benefit from systemic treatment and to improve the individual risk-benefit ratio. But I think it comes all down to multi-stakeholder engagement is really needed, collaboration between all stakeholders that are here represented in the room to address all these challenges and to ensure a high adoption rate, and so that it not takes, again, 20 years until we have changed our paradigm completely how we define doses for our patients. Thank you.
We will not have an individual discussion now, but we move directly into the panel discussion. So Harald, if you move to your right, and may I ask the other speakers to come up. So first, we would like to introduce our guest panelist. Benoit Daniel is a medical oncologist working at the MHRA since 2009. So very welcome here to add the UK perspective to the discussions we have. Do you want to say something at the start?
I think I feel more like a student getting a front row seat with so many experts on the panel. So I'm learning more than I have to offer. So yeah, and what I felt was in a lot of the discussions, everyone was pointing towards the same thing. And I think the last slide, Harald mentioned about multiple stakeholder engagement working together. I think that's an important point just to take forward.
Thank you. This feeling as student is, you're not alone in that, I can say that. So just at the start, because we've, unfortunately, Hans Scheuer cannot be here for a reason beyond his control. Katrin and I thought that we make a very brief statement of what has come up in the discussions we have had with patient advocates. So I think a key point for patients is actually to look at the long-term toxicity, long-term tolerability, because the DRT paradigm does not allow to assess that. So I think patient's perspective is really, what is the drug causing? What are those severe, or let's say for the patient, severe drug reactions like fatigue, like nausea, etc., that are not well detected in the current system. So how can we get better there? The role of PROs, we will discuss that tomorrow as well. The overall quality of life, that is perhaps not yet fully weighted into the system and into dose selections. The other part that Hans made at previous workshops is also the pursuit of response at any price as the demonstration of drug efficacy, and the route to accelerated approval has a danger in itself that high responses may not translate into the same optimum overall survival. So that should be studied and should be kept in mind. And I think there are a lot of examples. Philip, you mentioned bortezomib, which was also, from my experience in the clinic, very, very effective drug, but then neuropathy would kick in, and later we have seen a very beneficial reduction of the dose of the drug, which actually prolonged treatment duration and a longer clinical benefit. So that is a very good example, I think, that we should keep in mind. And I'll turn it over to Katrin here. And you have an eye on the online part.
Yes, should have. So we are working on getting the online questions back. But there was one question that was open for quite a while, and that was related to Mark's presentation. And I'm starting with this online question first, and we can also go to the room. Mark, there was a question. You presented that the FDA or even the US government is looking into, insurers potentially, or the NCI sponsoring those optimization studies post-approval. Can you elaborate a little bit on this? How would this look like? Who would be paying for this? Would that be an alternative to do this before? So give us a little bit of thoughts around, the post-approval determination and running of these post-approval trials.
I think what we have written about and what the US Senate has written about relates to post-marketing dose optimization studies. And the reason that the US government and other payers, and I would add that the UK NHS is also interested in this strategy, is that if one does a randomized trial where half the patients receive a significantly lower dose, the savings on the drug from 50% of the patients more than covers the cost of actually running the trial. And when I discussed this with the NHS a few years ago, their response was, we don't mind really large trials. In other words, they figured out it actually has a negative cost. And so payers can do this if they have the legal authority and the political will to do it. And I think the point is, if we're optimizing doses before approval, well, that's wonderful, but it doesn't fix the existing problems. And the same principles apply to drugs that we failed to optimize as to those that we have not yet optimized. So I think that's the opportunity. How it actually works is above my pay grade, because I think there are legal barriers that could be changed, obviously. Legislatures can pass laws enabling such trials or even directing such trials. And so I personally work with my elected representatives in this context to try and make change in the United States. But of course, every country has its own issues.
There's one more, that we start with the online questions. And I think there's one for Elena. We focus very much on the starting dose, and then potential, that's what we discussed mainly now in the presentations. But one of the online questions is, what about different dosing regimens? So could we discuss one dose, but different dosing regimens? Should we randomize to different dosing regimens rather than to different starting doses? What are your thoughts on this?
Well, thank you for the question. I mean, I think we need to consider dose, no? So dose and schedule, that is the dosage, no? Sometimes we think, at least when we're not the English speakers, we interchange it, no? But a dosage should have dose and schedule in between. And certainly, I mean, I think we are starting to see a lot of trials that are already incorporating dose, the schedule question in it. I think we still see less regarding dose, but schedule, we are seeing it. I think it's key, and I don't remember who mentioned it, to have the flexibility in the protocol to allow for this to happen without amendments, because this, of course, delays everything. And sometimes it's something that we just do it when we don't have any way out of it. But if we incorporate some language in the protocol and we incorporate this flexibility from the beginning, there's also going to be more willingness to do it from the beginning. It's sometimes if we are going to be forced to stop the trial for 3-4 months in order to explore another schedule, we just say, okay, it's not worth it. But if from the very beginning we include wording and we assume based on preclinical data, and even if not, we have the wording that allows us to study different schedules, I think we would all be willing to do it. It's just a matter of understanding that sometimes this flexibility needs to be written in the protocol from the beginning, and that the regulatories and ethics understand that behind this, there is a need that is going to be benefiting patients in the long run. It's not that right now there's not the actual knowledge, which is not a negative thing, but actually a positive thing. That I think sometimes we want to write protocols that are very structured and think that it's because we have everything clear and this is going to be more secure and for patients, but it's not in the long term. But yeah, definitely both things should need to be considered.
So the highest voted question in the online part is, and I fear to blow up the panel discussion with it, is about combination therapy. And if those of you willing to give an answer, keep it short. I think that will be addressed on a later workshop by the CDDF in the second half of the year. But we shouldn't shy away completely from it. If you kindly give a few thoughts, just keep it short. And Harald, you are bravely moving forward.
I can try. So I just wanted to share with you how we do that internally at CGEN. And we normally start as we usually do. We start with dose escalation, with establishing the safety and the tolerability and efficacy of monotherapy. Now that usually applies to our antibody-drug conjugates, which contain the cytotoxic agent. So I think it's largely advisable to do the monotherapy dose exploration first, and then later on, once the recommended phase 2 dose or doses or schedules have been defined, then there may be a plan to start combining it with other agents. Now, for non-cytotoxic agents, for biologics, targeted therapies, it may be possible, and it was done already, for example, for TIGIT inhibitors, where in parallel to dose escalation of monotherapy, combination was already started at 1 or 2 lower dose levels or dosing cohorts. So it's doable, but obviously it increases the complexity enormously.
Anyone else wishing to add? Philip?
Yeah, to add to that, I think that from our point of view, there would be 2 different scenarios of combination therapies. The one would be the add-on to an already registered regimen, where presumably in most cases, that regimen would be sort of the reference and would be the backbone for the add-on. I think that's what we usually see, rather than studies where you have an add-on to a backbone, where the backbone is the dosing is modulated. I would think that's less common. Otherwise, you end up in the situation when there's 2 or more new agents. I think I would concur that in most cases, a description of the exposure response for each agent would need to be the first step.
Thank you.
Can I? Perhaps just to mention very briefly that when we are developing combinations, toxicity is probably one of the main problems that we have. So I think that is why this also remains very relevant. I mean, because toxicity is really one of the highest challenges when we are developing these combinations, and why a lot of them fail.
Yeah, I think just in terms of what was discussed, I think what was discussed for monotherapy previously as well, the PK data informing what the toxicity might be, how the drugs interact. So we have that data also that might inform how the doses could be combined in the combination to be studied. So that is also important to look into. That's just one of the thoughts I had.
And Mark?
Yeah, I think we need to stop being toxicity focused. There's no a priori reason to believe that a drug that an effective dose of a drug requires that the patient experience toxicity. Okay. And I think once we start to believe that, we'll be a lot further along. And whether we need to look at other therapeutic areas to better understand why this is true, Other therapeutic areas do not talk about MTD. Take the anti-cancer drug nintedanib. How many pulmonologists know what the MTD of that drug is? It's approved for both oncology and IPF. It's just not a concept outside of oncology. Drugs have toxicities in many patients, and we like to avoid them. But we just don't talk about it. And so I think we have to get away from that. We have to get away from the idea that we have to be perfect. I think, I know it's controversial who first said perfect is the enemy of good, but I think we need to remember that. And we need to stop worrying about precision, about getting things exactly right. I think we want to study large range of doses. If the potential range of doses to be studied is small, there's probably not a large point in studying it.
Thank you. There's a question here.
Yeah, just Philip, as
If you can identify yourself.
Yeah, I'm Astrid Schott from Berlin, Germany. If the backbone therapy, we add on a novel treatment, usually it's not the best biologic dose because it hasn't taken into consideration, these new rules. Many things are just brought up to the biggest dose. So do you think that, when you look at this, you should also integrate different dosages of a backbone regimen to really find the best biologic dose? Because that is going to drive costs and time up enormously, but might lead to a better treatment.
Yeah, I think this gets down to what you have in it, because I guess we're talking about registrational pivotal trials with statistical power to support approval. And I guess one of the challenges here is that most of these tests, what you could call, they don't test a single dose. They test a treatment strategy, including a dose reduction scheme. It might be instructions about dose interruptions. There could be instructions about the duration of therapy for different element, different components, and so forth. And ultimately, in combination therapy, you may come up with a very large number of different parameters that could be studied in a pivotal trial. But of course, you have sort of the constraints to this, and again, I don't think we see a lot of studies where there's a lot of manipulations on the backbone when that's already approved. I think it's probably more about getting the add-on drug right. Though there are, of course, examples where there's PK interactions, there's such where there needs to be, but then the adaptation is usually to meet the same exposure that was registered anyway. But I guess it comes down to what are the key research questions, because I think even if you do a pivotal trial with, say that you have two different regimens you're testing against your established control, what we usually see is that the sponsor will split alpha between, they'll test the one regimen against the reference and the other against the reference. And then statistics with respect to the differences between the two, schedules or regimens or posologies, that will be more exploratory. That the study won't be powered sort of to detect that. I think that's, and I think in many, we've had studies like that, and we tend to, very simplistically as regulators, do they win statistically? Well, then they're approvable. If they don't win, they're not. And then, for instance, whether the add-on of an extra agent really matter might not really be elucidated. But that's just a limitation of the sample size, I guess.
Thank you.
Yeah.
So there is a common theme in the online question, and that's related to underdosing and exposing more patients now to potentially minimal not active doses by randomizing, going lower, to explore the minimal active dose, like we were discussing, randomizing more patients to these doses. And also, if you expose them to lower doses, is there a risk to introduce more resistance early on to the treatment? So there seems some concerns coming around here about exposing patients to a non-active dose or in low doses driving potential resistance to treatment faster. So any thoughts on this? Mark?
But I mean, no one is suggesting to expand inactive dose levels. I've never heard that suggested. I think once you see an efficacy signal, and again, we could go back to any of my favorite examples. Sotorasib is probably my favorite example because it looks to me from certainly at the first dose level, there was a partial response. Even looks like the first patient had a partial response at a dose of 180, which was predicted to be effective in the in the protocol. So once you see that your preclinical prediction was correct, well, you certainly wouldn't go up. You would certainly expand that dose level. You might think about going down because your preclinical prediction was correct. And it turns out that the 180 milligram starting dose wasn't ideal for a number of reasons because they didn't have a 180 milligram formulation. They had 30s and 120s. And so, but no one's saying, oh, well, at the first dose level, we need to put on 12 patients to try and find out if it's the minimally effective dose. And so I don't think there's an ethical issue here. I think in the context of a randomized dose ranging trial, one can, if one uses, for example, the endpoint that I've been suggesting, which is time to tumor growth, there's no reason that patients can't be escalated at any time where their tumor has gotten worse to see, if you can overcome that. And remember, these are drugs that eventually are going to have to be subjected to placebo-controlled trials otherwise in a regulatory context. And so I don't really see an ethical problem with that. I think it's unethical to administer unnecessary drug that causes toxicities that can even be fatal. So I think that's unethical.
Any other points of view?
I think, sorry. So yeah, if I can add, I mean, I think it's two things. No one wants to give doses that are subtherapeutic. I think it's a concern for all, and I think it's two different things. So where do you start? How you escalate quickly until you reach doses where you believe you're going to have an efficacy? I mean, that's a discussion in phase 1 design already. And another is dose optimization, when we are talking about doses that you are expecting to be efficacious already, and you have already some evidence of that. So I mean, for me, it's two different discussions.
I guess what I was alluding to in my lecture was that to completely characterize an exposure response curve, you need to give doses you assume are on the slope. And those would often be considered at least potentially suboptimal. And then I guess the consequences of exposure to suboptimal drug levels. My own research work was done on HIV, where the consequences of such exposure were devastating at times in terms of drug resistance. So I guess it both depends on the consequences of suboptimal drug exposure on the tumor and the clinical situation. Can you wait for the optimal treatment or not in that given niche? I would guess.
Yeah, if I can elaborate on that a little bit. We've seen these graphs of dose selection. So assuming that the minimal biological active dose was dose level 2, and the MTD was, what was it, dose level 7, and then you would go to a randomized trial with these two dose levels, and maybe or maybe not with a dose in between. But I think, we are not sure that this kind of design would actually find the optimal dose, because at the dose level 2, we may see some responses, but not all the responses that we can find. And MTD may again be too high for off-level toxicity. So does it mean that we always have to go to more than 2 arms with this selection strategy, more than 2 arms for the dose comparison? Are we, I think there is considerable uncertainty about this lower level. Not that it is totally ineffective, but you know, maybe the true, in quotation marks, response rate is 60%, and you see 30% at that level. That would be an underdosing. And so if you carry that on into randomization, I think in some cases that may be under treatment then.
There are published adaptive Bayesian dose response designs that have been used in other therapeutic areas. And so I think you can guard against that. I think one can reasonably say with the total number of patients one would like to, you know, study dose response in and then randomize them across to the dose levels that provide the maximum value of the information. And I think that's a general strategy. So that's what I would suggest. And I think it depends on the magnitude of the range. If you're looking at a 30-fold dose range, like was done with nivolumab, I think you want more than 2 dose levels. Again, that won't tell you what the optimal dose is, because in fact it's flat. We don't know what the optimal dose is when it's flat. We don't necessarily need to find that out in a randomized phase 2. You might find that out when you go to later, when you add it to a backbone where where one cohort of patients is going to get no active drug. At that context, it is reasonable to perhaps look at doses less than those that have been previously studied as well.
Just to comment on nivolumab, because it's an interesting case, because as I remember it, there were actually randomized dose-ranging trials for not only for renal cell cancer, but also for melanoma and non-small cell lung cancer. And in non-small cell lung cancer, the exposure, the dose response was not flat. Whether that was due to a too small sample or whether it genuinely was simply not flat in that less sort of immune sensitive disease, I guess we don't know. But I think that's an interesting case.
Actually, when you look at some of the FDA publications and they did a more robust analysis, they concluded that there was no dose response. And so if you email me, I can send you that reference.
I'll gladly do that.
Yeah, we have a question in the room.
Hi, this is Burak from Merck, Germany. So I have a question about the timing of doing dose optimization within the clinical drug development, because there is the potential issue of doing dose optimization before establishing the clinical effectiveness. And because we know that most of the drugs that in the phase 1 study will not be launched, right? The failure rate is high. So do you see this is an early optimization is an issue? And is the answer, already backfill cohorts were mentioned, do you think the answer to this is considering clinical effectiveness and safety in dose escalation? So changing the dose escalation trials, actually.
I would guess, and this is just right out of my heart, I would guess that post-authorization dose optimization would be based on a communal experience that the dose is not optimal. So in a sense, you could see the need for post-authorization dose optimization as incompleteness or even failure of pre sort of approval dose optimization. So I guess that the ambition would need to be to get the dose right for the first approval. I have difficulty seeing anything else.
Yeah, we have another question in the room.
Thank you. I'm Divya from Genentech Roche. Such insightful presentations from all the speakers, but my question is specifically for Harold. You nicely identified some of the study design complexities when it comes to the development of optimized cancer therapeutics. One such complexity would be if you have in a phase 1 study a heterogeneous patient population with different cancer types. In such cases, what is an efficient sequence for dose optimization? Would you optimize the patient population first and then go for more detailed dose optimization in that patient population? Or given that the minimal active doses could be different, would you start with dose optimization prior to the patient population?
Yeah, thank you. That's a great question that we are dealing with every day. So our preference, and again, this is mainly for ADCs, our preference has been, and experience has also been quite positive, to do the dosing and scheduling optimization first. And because, I mean, it depends, of course, also, is it all solid tumors? Is it a more immunogenic solid tumor? For blood cancers, in hematology, the situation is, of course, very different. But once we have a dose and a schedule identified or two, and then we see also what has been the preliminary efficacy, when has the efficacy occurred, we take the PK and biomarkers into account, and we try to do the indication selection thereafter. So dose and schedule first, indication later. Which doesn't mean that this is a one-size-fits-all approach.
The second highest, if we may just, one more online question. Second highest voted question there is what to do if there is not a biomarker. And I suppose biomarker means here a clear efficacy target engagement biomarker. How to drive dose optimization in that case?
Well, let me comment. I think people forget tumor size is a biomarker. So we have a good biomarker. We have a better biomarker than in just, in every therapeutic area other than antiviral, anti-cholesterol, and hypertension and diabetes. So much better than neurosciences or cardiovascular or pulmonary therapeutics. We've got a great biomarker. We've underutilized it. We get all these great tumor size measurements, and what do? We say they either responded or they didn't or they progressed. So we've got to start thinking more scientifically about this biomarker.
So with apologies to Jaap, out of the RECIST box, as you said, just looking at the net change in tumor size or ctDNA levels to look at the tumor response, not just in the categories of PR.
Is that truly outside of the RECIST box? I mean, Mark mentioned before that time to tumor growth is also an important biomarker. And I can remind you of, I was involved in the first imatinib study in GIST. Half of the patients that responded had no objective tumor regression. They had stable disease. And for some patients, this goes on for 22 years already. So looking at the kinetics of a tumor in the individual patient is just as important. And you can use RECIST measurements for that. That's not a problem. But looking at it and understanding it, I think that's important. We're starting to discuss here about response. Stable disease that is long-lasting is also a response.
In a randomized trial.
Sure, but also in a non-randomized trials, it's an indication there is something ongoing. There might be something ongoing, and certainly if you have you developed yourself the TTP2 versus TTP1 concept, so time to tumor progression on the new treatment versus the one on the previous treatment in the patient. If that's longer than 1, the ratio, there's something ongoing there because usually it's shorter than 1.
Yeah, I don't like that. Yeah, I hear you. But look, you are correct. If you see stable disease for a year, in a disease that progresses, that's most likely a drug effect. But when you hear people talk about, 75% of our patients had stable disease as their best or better, it's their best response, and they had no partial responses, what it means is they probably selected very indolent cancers for their clinical trial, unless you clearly have some patients with some tumor regression. I mean, sorafenib, as you know, has a very low partial response rate, but yet there was clear evidence of tumor regression.
I absolutely agree with you. And doing phase 1 studies looking for stable disease for 12 weeks to me is a total nonsense. So it should be a longer period. I don't think a year is necessary. It's also depending on the disease and the prior history of the disease in individual patients.
Yeah, there's actually also another question which is quite relevant for the discussion. What do we do if we have, very rare tumor types? And one of the questions was related, can we do randomized dose-finding studies in pediatric oncology? Any thoughts?
Well, I think it depends. My thought would be, is this an academic question or a pharmaceutical sponsor question?
It's anonymous.
Yeah, if it's coming from a sponsor, I'd say, well, there must be enough patients you're interested in commercializing the drug for this indication. If there are, there's enough patients to study it properly. If it's an academic question, it's an academic question. And then in academics, we learn what we can learn with the resources that we have.
Okay. Yeah, you have another.
Perhaps a question, Stefan Braun from Zug, Switzerland. Perhaps a question to all the panelists that now are advocating the efficacy or the tumor as a biomarker. So usually, unless I'm mistaken, most of our oncology drugs were registered for a indication, perhaps two, right? But we are supposed to enroll a mix of patients. How does that, so different indications, how does that fit together? So does that mean in the past we simply omitted tons of patients that should have benefited from the treatment? Or why are we selecting then one indication? But now in the phase 1, we say, let's register 20 indications and we get a good indication of the efficacy of the drug. Can you shed a little bit more light into how you do that practically in the protocol? And what is the practical experience that such a broad mix gives you the optimized dose for one indication later?
Yeah, thanks for the great question. So the selection of the indications or the tumor types for dose escalation is, of course, also based on some biological rationale. So we for example, we use TCGA expression data if there's a target, biomarker-driven indication selection, so that we usually end up with a mixture of 4 or 5 indications, maybe 6. So we have limited the number of indications already quite a bit before starting the dose escalation or the dose finding activities. And then later on, we try to continue reducing the number of indications based on what we see in terms of the overall evidence or the combined exposure response relationship.
But only a quick comment, absolute get it, but that means we take a decision already before we start escalating the dose, which we have not done in the past.
Agree, completely agree. So it's basically in terms of indication, and maybe I have to correct my previous answer, we do the indication selection in 2 steps.
Okay. I think we have time for one more question, and we have one more question here in the room.
Yes, thank you very much. My name is Susan Batty from Merck in the Netherlands. And I have a question about the suggestion of conducting post-marketing dose optimization studies. My feeling is it might be very difficult to recruit patients into those studies. It's quite difficult to recruit patients anyway, but to ask them to go back with an approved drug and try a different dose house and it might work, I think would be challenging. Whereas I've seen from what you've presented that actually physicians will reduce dosage anyway if they feel that the patient is not tolerating the drug. Wouldn't the better approach be to then capture that data in terms of real-world evidence and use that to optimize the dose rather than trying to set up new studies?
I think the problem with that is that it's a selected group of patients that gets reduced. So its patients with decreased drug clearance or who the physician deems are likely to have more toxicities. I would say actually it's quite feasible to conduct randomized dose ranging post-marketing trials. In fact, the FDA has required that of some companies, and Amgen has completed very rapidly a post-marketing trial of sotorasib comparing a 240 to their labeled dose of 960. We conducted a trial of abiraterone, 250 versus 1,000 without a problem and with virtually no funding. And there has to be a reason why patients would want to enroll in such trials. And it's usually because the labeled dose has toxicities or has to be given too often or in an inconvenient manner, such as with the abiraterone, patients were told they had to abstain from food in order to take their medicine. And we just said, give a fraction of the labeled dose with food, and patients happily went on the trial. The other thing is, in countries like the United States and Singapore, patients have some skin in the game and actually may have significant costs for the drugs themselves. So there's lots of approaches to conducting these trials.
Can I give you the last word?
Yeah, I have nothing. I think this answer was very close to what I was intending to answer. I think this is a problem that in order to inform your exposure response model, the studies where the data are derived must be designed in an appropriate way. And your proposal will result in the kind of confounding that I think Mark described very well here.
So we conclude here because one of the hardest rules in CDDF workshops is not to shorten the coffee break. So thank you very much. We know there are a lot of online questions we haven't answered yet. We have transferred a few to the other sessions. Otherwise, please ask again. Obviously, the question about Bayesian designs, randomized phase 2 statistical designs could be very well placed in session 3, for example. So we hope that over the time of the workshop, we will answer all your questions. But for the first session, many thanks to the speakers, many thanks to all of you.
And we will see each other 10 minutes before 4 o'clock in the room again. Thank you.